Thank you, everybody, for joining us for this symposium today. I'm going to start out by speaking about one potential pathway from poverty to psychopathology through the potential mediating role of hormones and the hippocampus. And I want to just take a minute at the start to thank all of my collaborators on this project, including Dr. Joan Luby, who is the co-PI of the grant that funded this work. I also want to take a minute to acknowledge disclosures and the generous funders of this research and to indicate that I don't have any conflicts of interest. So there's very strong evidence that early experiences of poverty, as well as other forms of adversity, contribute to an increased risk for poor developmental outcomes, including mental health problems. And we know that there's some evidence also that early adversity contributes to difficulties with emotion regulation, and that disruptions in emotion regulation in turn might contribute to risk for depression. But the question really is, what is the pathway or mechanism by which poverty is contributing to such deficits in emotion dysregulation and depression? And some people have really framed this in terms of, how does poverty get into the brain, or how does adversity get into the brain? We have evidence already that one way that adversity gets into the brain is through having an impact on the structure and function of the hippocampus, including reduced hippocampal volume associated with poverty, reduced maternal support, and other forms of abuse or adverse childhood experiences. And we know that this is really consistent with the animal literature that also shows the kind of effects of stress and environmental factors on hippocampal cell proliferation and things like dendritic length and branching and neurogenesis in the hippocampus, where there's a negative impact of stress and environmental poverty on these things in the hippocampus. I think we also know that the hippocampus is really important for stress reactivity and effective emotion regulation, which is one of the reasons why it might contribute to this pathway. What I think we've known less about is potential hormonal influences that might also be part of this pathway. So we know in part from the animal work that hippocampal disruption occurs through dysregulation of the hypothalamic pituitary adrenal axis, or the HPA axis. We know that chronic stress leads to heightened responses to stress and glucocorticoid secretion, which can disrupt hippocampal development. And we know this now from both human and animal studies. Interestingly, the HPA axis, though, is also involved in the onset of adrenarche and the release of hormones like DHEA and testosterone, which are really important for brain development. We know that higher levels of testosterone and DHEA from animal models can suppress cortisol secretion and help regulate stress and the excitotoxic effects of stress on stress reactivity and cortisol release. And there's also evidence that androgens, like testosterone in particular, support neurogenesis and cell proliferation in the hippocampus, at least in adult rodents. So this raises the intriguing possibility that another part of this pathway from poverty and early adversity to emotion dysregulation and depression through the hippocampus might involve modulation of testosterone release across development. To test this, we had the good fortune to have data available from a prospective longitudinal study referred to as the preschool depression study. So this study started when children were in preschool, as it involved multiple assessments over the course of development into late adolescence. We had measures of poverty and depression and other early adversity factors early in life, including in preschool. And then we started to measure brain structure over the course of development, as well as to measure hormones like testosterone and DHEA over the course of childhood and early adolescence. We also have measures of depression and emotion regulation from later on in children's development. And so this study design allowed us to look at early factors that would have an influence on brain development and behavior over time, including early poverty, allowed us to assess hormones like testosterone and to look at outcomes like depression and emotion regulation when youth were in their kind of early adolescent years. Now, the relationships that we wanted to look at was whether or not early poverty, as measured by income to needs, which is considered to be a good measure of early poverty, related to these outcomes at later points in time. So T1 refers to the preschool era, and T9 refers to adolescence, when children were approximately age 16. And these testosterone and hippocampal measurements occurred over time from when youth were in school age through adolescence. And we were able to look at the slopes of testosterone and hippocampal change over time. So we first started by looking at whether poverty related to these factors in the way that we would predict from the previous human and animal literature. And indeed, we see that a positive relationship between income to needs and testosterone slope, such that youth who had higher levels of income, so lower poverty when they were at preschool, had higher slopes of testosterone increase over the course of development. We also saw that income to needs was related positively to hippocampal slope. So children with more income had steeper growth of hippocampal slope over time. And we also saw that early poverty related to better emotion regulation when they were adolescents. So higher poverty was related negatively to emotion dysregulation. And we also saw that early income to needs was related to adolescent depression, such that youth who had higher poverty early in life had greater depression when they were adolescents. So we first confirmed these basic relationships between poverty and these other factors. We then looked at how testosterone slopes related to these other factors. And interestingly, we see a positive relationship between the increase in testosterone over the course of development and increase in hippocampal slope over the course of development, such that youth that showed a greater increase in testosterone showed a greater increase in hippocampal volume over time. We also saw that testosterone slope was negatively correlated with emotion dysregulation, such that youth who showed a greater increase over development showed less emotion dysregulation when they were adolescents, as well as less depression when they were adolescents. We then looked at the relationship of hippocampal slope to these factors. And similar to previous research, we saw that a greater increase in hippocampal slope over time in development was associated with less emotion dysregulation when youth were adolescents, as well as less depression when youth were adolescents, confirming again previous relationships. Now, the critical question, though, is really a mediation one, and whether or not there's evidence that there is a pathway between early poverty and later depression that is mediated through this pathway through testosterone, hippocampal development, to emotion dysregulation, to depression. So to test that, we used process models to test serial mediation. And the first model that we tested was a serial mediation model asking whether there was a pathway that significantly accounted for at least part of the variance between poverty and depression that went through testosterone, hippocampal slope, and emotion dysregulation. And consistent with this hypothesis, as illustrated in green, we saw a significant indirect pathway from poverty through testosterone slope, through hippocampal volume, to emotion dysregulation, to depression. Critically, this is true even if we controlled for levels of depression when youth were in preschool. If we flipped the direction of this pathway such that, for example, we put hippocampal slope before testosterone slope, we do not see a significant indirect pathway. It is also the case that if we look at emotion dysregulation slightly earlier in adolescence, prior to the depression, we also see a significant serial mediation model. So while this, of course, does not provide causal evidence, it is consistent with the idea that one part of the pathway by which poverty may influence depression later in life is through modulation of testosterone secretion over the course of development, which in turn may relate to hippocampal growth, which in turn may contribute to emotion regulation or dysregulation. So in summary, our findings are consistent with the hypothesis that early poverty contributes to disruptions in the HPG axis function. Ideally, to really test fully this model, we'd also have measures of cortisol across the course of development. So we could measure hypothalamic pituitary adrenal axis function. So we can have a better sense of whether that's part of the pathway as well, because we would be hypothesizing that cortisol would be influencing HPG axis function. I should note that there is some evidence in the literature of a positive coupling between cortisol and testosterone in adolescence in the context of adversity, which would be really the opposite of what we're predicting here. But there may be important differences depending on the time scale of assessment. Those previous studies on a positive coupling have really been within the course of a day, whereas we're looking at the relationships between hormones and brain development over the course of development across the course of many years. I should also note that this work is consistent with prior research suggesting that testosterone may have beneficial effects on hippocampal development, including evidence for a positive relationship in animal models between testosterone levels and neurogenesis and cell proliferation, particularly cell proliferation in the hippocampus. And this is really consistent with human research also looking at levels of androgens and brain development, which also suggests a positive relationship between testosterone and hippocampal volume, as well as some other brain regions. Now, I should note that prior research has primarily been cross-sectional, and even though our work is longitudinal, it doesn't establish causality because we are still looking at sort of naturally occurring relationships. And so it will be important, for example, to bring in additional animal models where we can more experimentally test these hypotheses. I should note, however, that we did look at other control regions like the amygdala, the caudate, the dorsal anterior cingulate, the dorsolateral prefrontal cortex, and we didn't see evidence for similar relationships there. So this does suggest some level of specificity to the hippocampus, which again is interesting in the context of the role of the hippocampus in stress reactivity and emotion regulation. Thank you.
Hi, my name is Dr. Dylan G. I'm from Yale University, and thank you for the opportunity to share some of our recent work on early adversity and brain development. Experiences during childhood play a crucial role in shaping the developing brain, behavior, and risk for psychopathology. A nuanced understanding of how early experiences alter brain development is critical to elucidating the mechanisms by which childhood adversity confers risk for psychopathology and how protective environmental factors may buffer that risk. Across species, a growing literature has identified alterations in structural brain features such as gray matter volume, cortical thickness, white matter tract integrity, and myelination following adversity. While such evidence has been foundational in establishing the effects of early childhood environments, there's vast heterogeneity in both the nature of adverse exposures and in outcomes. And the frequent co-occurrence of traumatic experiences and additional complexity of family, neighborhood, and school environments present further challenges to precisely linking environmental factors with variation in brain structure. Data-driven approaches may be particularly helpful for addressing these challenges. In the current study, we aim to decompose heterogeneous associations between brain structure and specific aspects of the childhood environment in a sample of youth from the Adolescent Brain Cognitive Development, or ABCD study. We leveraged a novel multimodal data integration framework called Similarity Network Fusion, or SNF. SNF was originally developed to integrate multimodal data in the genetic field and has recently been applied to neuroimaging studies. Compared to traditional unimodal approaches, this framework allowed us to simultaneously take into account environmental factors and structural brain features in clustering. Brain structure was examined using cortical thickness and a myelin surrogate marker of T1 weighted over T2 weighted ratio, and environmental factors were selected to characterize youth environment with regard to trauma exposure and family, neighborhood, and school environments. Here I'll walk you through the approach that we used for subtyping. First, the brain structural features and environmental factors were used to create a between-subject similarity network at each modality. The resulting similarity graphs were then entered into SNF, which performs iterative nonlinear fusion processes to combine them, resulting in a single fused similarity network. Spectral clustering was then applied to find homogeneous subgroups. To find the most reproducible clustering results, we bootstrapped 90% of the samples and repeated the SNF subtyping. This procedure was iterated 1,000 times, and the subtype results were aggregated to construct a consensus clustering matrix. The final clustering result was obtained based on spectral clustering of this consensus matrix. And importantly, this entire subtyping process was performed 2 separate times, once for the discovery data set and once for the replication data set, in order to independently assess the reproducibility of our findings. To assess the significance of identified subtypes, we conducted prediction analyses of subtype membership classification and of clinical symptoms. Results of similarity network fusion revealed two clustering solutions, one with 2 subtypes and one with 5 subtypes. First, I'm presenting results of the 2 subtype solution. And as you can see here, the 2 subtypes significantly differed on both metrics of brain structure with inverse patterns of cortical thickness and myelination. Individuals in subtype 1 versus subtype 2 differed in the extent to which their environments were characterized by parental and caregiver support and by neighborhood safety. Specifically, individuals in subtype 1, shown here in blue, were characterized by reduced cortical thickness and increased myelination, and had higher support displayed by parents and caregivers and lower perceptions of neighborhood safety than individuals in subtype 2, shown here in red, who were characterized by increased cortical thickness and reduced myelination. The 5 subtype solution also identified subtypes with robust differences in cortical thickness and myelination. Consistent with a more fine-grained parsing, there was greater variability in brain imaging and environmental phenotypic profiles between the subtypes. Though there are many patterns of co-occurring brain and environmental factors to unpack here, I will focus on a couple of robust patterns. Similar to the 2 subtype solution, you can see here that subtype 1 was characterized by higher myelination and increased parental and caregiver support, shown here on the right in blue. Another notable association was observed in subtype 3, shown on the right in yellow, where relative cortical thickening co-occurred with less favorable conditions in almost every domain of environmental factors, including lower parental and caregiver support, lower parental monitoring, a more significant family history of mental health problems, less favorable school environment, and higher family conflict. Importantly, the majority of these findings in both the 2 subtype and 5 subtype solutions were reproduced in the independent replication dataset. Our first prediction analysis assessed how generalizable the brain structural and environmental phenotypic profiles for each subtype were in unseen cases through subtype classification. Classification accuracy was 86% for the 2 subtype solution, where chance level would be 48%, suggesting that the brain structural and environmental profiles learned from the discovery dataset for each subtype were highly generalizable. Although classification accuracy was lower, generalizable subtype-dependent profiles were also found in the 5 subtype solution, which revealed successful classification in 63% of cases where chance level would be 19%. In the second prediction analysis, we attempted to predict clinical symptoms based on externalizing, internalizing, and total problem scores from the child behavior checklist with brain imaging features in unseen cases. The prediction of clinical scores based on support vector regression showed marginal but significant correlations between the actual and predicted scores. And the classifier informed by the 2 subtype solution successfully predicted externalizing symptoms, but not internalizing symptoms or total problems. And the classifier informed by the 5 subtype solution successfully predicted total problem scores, but not internalizing or externalizing symptoms alone. On the right, you can see the features that contributed to the prediction of both classification as well as clinical symptoms, which were primarily located in lower level sensory motor and higher order default mode areas, suggesting that these regions may be particularly sensitive to the environment during childhood in ways that are relevant for mental health. Using SNF, we identified consistent patterns of co-occurrence between brain structure and features of the childhood environment. Among these patterns, increased cortical thickness was commonly observed in conjunction with less supportive caregiving, higher family conflict and psychopathology, and higher perceived neighborhood safety. Increased myelination was most consistently observed in the context of more supportive caregiving and school environments. And I would encourage you to check out our preprint on bioRxiv for additional information, including regionally specific findings. More broadly, our findings support the idea that distinct environmental exposures are differentially associated with brain structure during childhood. Taken together, our findings suggest that this type of data-driven approach may enhance understanding of how environmental and brain structural variation co-occur, Notably, the subtypes that we identified here were highly reproducible across the discovery and testing data sets. And these findings also highlight the importance of a multimodal approach, as SNF findings based on either brain structure or environmental factors alone did not yield comparable subtype profiles. The prediction analyses that we conducted in unseen cases further suggest that this approach to subtype classification may also have clinical relevance. Importantly, when we performed the prediction of clinical symptoms across all participants without subtype information, we failed to predict any clinical symptom scores. These results collectively emphasize that given a highly heterogeneous sample, identifying more homogeneous subgroups may be a meaningful step to take before examining associations between brain structure and phenotypic data. In the future, delineating more precise associations between risk factors, protective factors in the environment, and brain development may help with early identification of risk as well as optimizing interventions based on profiles of adversity and neurodevelopment. I'd like to thank the many people who have contributed to this work, including my co-authors and especially Lucinda Sisk at Yale and Sukjun Hong at the Child Mind Institute, who co-led this work. I'd also like to thank the ABCD Study Consortium and all of the families who participated in the study, as well as our funding sources. And thank you for your attention.
Okay, I'm going to talk today about the dimensional model of adversity and psychopathology. So childhood adversity encompasses any exposure to which requires significant adaptation on the part of the child and which is quite negative. And there is actually an enormous body of research linking exposure to adversity in childhood with long-term deficits in health and well-being. And this data not only links to health and well-being, but also shows relationships between adversity exposure and changes in the brain and biology as a result, which are long-term. And many of these associations have been confirmed through randomized trials where kids who are randomly exposed through natural experiments or randomly sampled out of adversity exposures show differences in long-term differences in biology and also health outcomes. One of the questions which remains unanswered is how adversity gets under the skin to affect health and well-being. And one of the most common approaches for understanding this association to date is the adverse childhood experiences approach, which is a cumulative risk approach where the number of exposures to which a child has experienced are linked with negative outcomes, all forms of negative outcomes later. And the primary mechanism hypothesized to link these two things is a stress mechanism where increased exposures results in increased stress and then thus deficits. And that's a really important mechanism in this particular way of linking childhood experiences to negative outcomes is an important way of identifying who is at risk and has been very important and productive in that way. But one of the things that's really missing from this conceptualization of childhood experiences is the idea of developmental plasticity, the idea that in early childhood, brains are really designed to be taking input in from the environment and then using that input to design the most efficient brain for the future of that child. And in response to this gap in the literature, my colleague Kate Mclaughlin and I developed the dimensional model of adversity and psychopathology. And here we're really identifying dimensions of exposure that go across specific exposures. And we think those dimensions will selectively be linked with negative outcomes later. So on the x-axis here, we have our first dimension, the idea of threat, which is exposure to interpersonal violence in the community or in the home. And the idea is that threat exposure is not a species expected learning experience. So it's a novel learning experience that the brain will incorporate to prepare itself for future experiences of threat. And so what we expect to see are changes in the parts of the brain linked with fear learning and salience processing, increased emotional reactivity, and decreased emotion regulation. And specifically here, we're thinking the amygdala, hippocampus, and medial prefrontal cortex should be most linked with threat exposure. In contrast, we think of deprivation as the lack of species expected learning experiences. So here, this is a very different, well, it's a similar mechanism, but a different idea about what's going to happen. So the lack of exposure to these expected cognitive and social stimulations, because we know the brain requires input from the environment for early normal development, we think that if these exposures are absent, we expect that the connections that process those kinds of information will be eliminated at a greater rate or in increased way, and that this typical developmental process of synaptic pruning will be hijacked by deprivation, resulting in cognitive deficits and fewer connections, specifically in association cortex, which we think of as the area which is where those expected inputs would be processed. So things like the dorsolateral and ventrolateral prefrontal cortex and the parietal cortex. And we were basing this early idea on work that we had read, early work we'd read in animals and in humans looking at sensory deprivation. So the idea that if you deprive humans or animals of visual stimulation early in development, then selectively in visual cortex, where those inputs would be processed, you see increased cortical thinning and decreased synapses. And we knew that there was other work that looked at more global forms of deprivations in animals and humans. And in that work, that more global deprivation sort of resulted in more general decreases in cortical thickness across more parts of the brain. So we took this general idea and we then tested it in a few initial studies in adolescents and young adults. And what we observed in those studies were that different dimensions of exposure did affect the brain behavior differently, and they were robust to controls for each other. So the general form here is that we're examining deprivation and threat exposure in the same regression. We're predicting an outcome which we think will be selectively linked to either threat or deprivation, and we're observing that selective association. And across a number of studies at this point, we have observed selective associations between deprivation exposure and things like inhibitory control, recruitment of the prefrontal cortex and parietal cortex to support working memory performance, changes in language development with deprivation, and with threat. In contrast, we've seen across a number of studies increases in emotional reactivity and decreases in automatic emotion regulation, and changes in neural recruitment, which reflect those differences. To date, however, there has been no work in early childhood, which is the moment we expect these associations to be emerging, because this is the time period where that early cognitive and social stimulation is most important. And I'm going to present two studies today that were both done by Laura Mclaughlin in my lab, who's a graduate student. She'll be on the job market in a few years, looking for a postdoc, not next year, but the year after that. So look out for her. So in the first study, this is data from a pilot study for the Wellness, Health, and Life Experiences study. There are 64 kids. They're about half female, typical IQ, and they're between the ages of 4 and 7. In this study, what we've done is we've identified deprivation using an index. So we have the child's own report of neglect, the parent's report of cognitive and social stimulation in the home, and parental education. For threat, we're using the child's report of violence exposure, the parent's report of interpartner violence, and then a questionnaire which assesses predilection for abuse in the parents. And all of our analyses are controlling for age, gender, and IQ. So here, first, we're going to test the relationship between threat and deprivation and cognitive outcomes that we expect to be linked to cognitive behavioral outcomes. So for cognitive control, we're using assignment task. It's a very simple task. It's a block design. Kids press on the same side when they see a pink circle and on the opposite side of the screen when they see a yellow circle. We're just going to compare their performance on incongruent blocks to congruent blocks to get an estimate of their cognitive control using their behavior. In the fear learning task that we gave kids, also very simple, also block design, we're presenting 3 kinds of stimuli: a CS minus, which here is just a blue square where they see the blue square and nothing happens; a CS plus, which has been reinforced, so that's here an orange diamond, where a loud sound is going to play on about 80% of the trials; and then also the CS plus non-reinforced, which is just an orange diamond where no sound is played, but it's during the same task where the diamond was previously paired with the sound. And here we're going to measure skin conductance as our outcome and look at skin conductance both to the reinforced trial, so just skin conductance to the loud sound, to the fear stimulus itself, and also skin conductance to the non-reinforced trials controlling for the CS minus. What we observe is that in fact deprivation predicts cognitive control. So this is inverse efficiency on incongruent trials. You can see as deprivation increases, inverse efficiency increases, so kids get worse at those trials. And this is controlling for congruent trials: threat, age, gender, and IQ. And we see that in contrast, threat was not related to cognitive control. We see that threat is related to skin conductance to the unconditioned stimulus. So this is just the fear stimulus itself. And what we see is that kids with more threat exposure have a more blunted response to the unconditioned stimulus, so that loud negative noise the kids heard. And this is controlling for deprivation, gender, age, and IQ. Really interestingly, if you look at the condition of interest here, the CS plus minus the CS minus, so this is the non-reinforced CS plus compared to the non-reinforced CS minus, you see the kids who are younger here on this part of the graph, kids between the ages of 5 and 6, show are better able to differentiate between those two kinds of stimulus and their risking conductance than kids who are not exposed to threat. So the high threat kids show an ability to differentiate and the low threat kids don't. Across time, across age, all kids show increases in their ability to distinguish between the two stimuli, but threat only distinguishes kids in those early ages, which is consistent with the idea that at earlier ages you would begin to distinguish between threat and safety cues if you were a kid who was exposed to threat in the home. Of course, this is a really small study that's driven by a couple of data points, so we're trying to replicate that in a larger study now. Now, the data I just showed you previously was really consistent with all of our hypotheses with the DMAP and replicated a lot of previous findings that we had in older kids, which was exciting, but none of it was related to the brain. So the data here is really looking at the impact of deprivation and threat on neural structure. And this is using the Duke Anxiety Study, which is a study of preschoolers who are at risk for anxiety. So about half the kids in the study, there are 68 of them, about half of them met criteria for anxiety disorder, and that's how they were selected into the study. But it happened that in the course of the study, they also measured a bunch of aspects of adversity exposure, and we're going to use those to examine deprivation and threat. And we initially looked at interactions with anxiety, but I won't be reporting on those today because none of that was significant, which was sort of a surprise. So here we're going to look at parent report of neglect exposure. And that's going to be our primary measure of deprivation. It was the only measure of deprivation that was available in the study. And we're going to look at parent report of domestic violence, abuse, and violent environment. So that same interpersonal violence variable that we looked at previously as our index of threat. And here we're going to be able to do something a little interesting, which is we're going to be able to control for cumulative risk. So this is really a more direct test of the idea that differentiating between deprivation and threat is a more effective way at identifying mechanisms of adversity exposure than just a cumulative risk or grouping approach. And to look at cumulative risk, we picked some events that were neither deprivation nor threat, but they were severe stressors that had happened in a child's life. Things like death of a parent, natural disasters, vehicle accidents, hospitalization of the child, or becoming homeless. So clearly stressful events, but just not events which would be classified as either deprivation or threat. And what we're going to do is we're going to examine neural structure in 3 different ways, and we're going to control for cumulative risk and the other dimension when examining our outcomes. So first, we're looking at subcortical regions of interest in the amygdala, hippocampus, and striatum. We're going to look at thickness in the whole brain and surface area in the whole brain, and we're going to look at cluster level corrections for, so we're going to look vertex by vertex, and then we're going to look at cluster level corrections in the whole brain. And what we see, so here we expect that the hippocampus and amygdala will be associated with threat, and we expect that thickness will be associated with deprivation, but not vice versa. We really didn't have hypotheses about the striatum or surface area, so striatum is here serving as kind of a control, showing that it's not all subcortical structures that are related. And surface area was really an exploratory approach. We didn't expect anything to be related to it, but we thought it would be a shame not to look at it, so we examined it here. So first of all, what we see is that threat is in fact related to amygdala and hippocampal volume. And you can see that more threat exposure on the x-axis is related to smaller volume of both the amygdala and the hippocampus. This is robust to controls for cumulative risk, anxiety, age, gender, and the scanner on which the data was acquired, as well as deprivation. Deprivation wasn't related to amygdala or hippocampus, and neither variable was related to the striatum. For cortical structure in the whole brain, here we're looking at surface area. And interestingly, what we observed is that threat very broadly across the whole brain predicted smaller surface area. And this is again robust to deprivation, cumulative risk, anxiety, age, gender, and scanner as controls. We really, when we first saw this finding, thought that the association was actually going to be that cumulative risk was related to surface area. So we introduced cumulative risk as a control, but we found that cumulative risk did not result in decreased surface area, and that the effect of threat on surface area was robust to that control. So this really does seem like an association with threat. If you reduce the p-value here at all, or you include fewer controls, you just see that this extends across the whole cortex, although the strongest associations are here in the prefrontal cortex. You can see both laterally and medially. And deprivation was not associated with surface area at all. In contrast, deprivation was associated with cortical thickness. It's stronger in the left hemisphere laterally, which we've also found in a few other studies. But you can see it extends through much of association cortex in the left hemisphere, as predicted, threat was not associated with cortical thickness. Interestingly, deprivation does not predict thinner cortex here, it predicts thinner cortex. It predicts thicker cortex. So that was counter to our predictions. So in conclusion, we do see that deprivation and threat are differentially associated with neural structure in early childhood, and this is even controlling for cumulative risk, which really does support the idea that these sorts of adversity exposures should be differentiated if you're taking a mechanistic approach to understanding the impact of childhood adversity on brain and behavioral development. We expected to see deprivation to be associated with cortical thickness, and we did observe that. We expected threat to be associated with subcortical volume, and we did observe that. We didn't expect deprivation to be associated with increased cortical thickness or threat to be associated with surface area. Now, in examining the literature that has happened more recently, since the time we initially looked at the sensory deprivation literature, it does look like in more recent human data, increased cortical thickness is associated with congenital blindness. So it could be that our initial understanding of the sensory deprivation literature was wrong, specifically with regards to cortical thickness, which is of course imperfectly related to synaptic pruning. And our initial hypotheses were also based on mature but not developing animal models, so situations in which those impacts happened in a mature model and not actually related to developmental plasticity. So it could also be that here in early childhood we expect thickness, and maybe that will eventually change into cortical thinning. And we actually will have an ability to observe that because it looks like we're going to get a chance to follow these participants up now that they're adolescents. These threat associations with surface area were very global as described, and in this data set and in early childhood, we also see a huge impact of threat exposure on child functioning. So this general associated with surface area might be related to that really large impact of threat exposure on kids' mental and physical health. And just sort of as an index of how really important threat exposure is, specifically in this early childhood period. And we also are hoping to follow up that finding with sort of other investigations of surface area, given that, see if we replicate that result and see if others replicate it too. So in conclusion, I'd just like to thank my collaborators, my students in my lab, and my funding sources, and thank you for your attention.
So thank you so much for having me here today, and thank you to the organizers of this symposium. So today my talk will focus on the neural mechanisms underlying the income achievement gap, with a particular focus on trying to understand the role that the ventral visual stream plays in explaining this gap. It is well established that children reared in low-income households go on to have lower levels of academic achievement than their higher income counterparts, and this is known as the income achievement gap. This is a figure from a study that came out last year in the United States. And what this line shows you is academic performance from the top and bottom deciles of the socioeconomic status spectrum. And what this shows you is that this gap in performance is large. It amounts to about 1 standard deviation of performance, which amounts to about 3 to 4 years of learning. And interestingly, this gap has not changed over the last 50 years. And moreover, this gap exists in the United States, but it's not unique to the United States. It exists across the world, including in countries with large social safety nets. Recent work, this is sort of a burgeoning field of trying to understand the relationship between socioeconomic status and brain structure and function. So recent work has focused on trying to understand differences in cortical thickness, differences in cortical surface area, and differences in functional activation during difficult working memory tasks, and whether these differences play an important role in academic achievement, in explaining this achievement gap. Many studies that have tried to uncover the neural mechanisms of the achievement gap, including work from our lab, have focused on the frontal parietal network. And this makes sense. The frontal parietal network is critically involved in executive function, a set of cognitive skills that are very important for academic achievement and that have also been linked to socioeconomic status. So indeed, recent work from our lab has found SES-related differences in functional activation in the middle frontal gyrus, in the superior longitudinal fasciculus that connects the parietal and prefrontal cortex, as well as cortical thickness in the MFG and the IPS. And we have also found that these are linked to academic achievement. Indeed, work from Amy Finn has found that functional activation in the frontal parietal network is associated with both socioeconomic status and with academic achievement. So all of this has been sort of critical to trying to understand the neural mechanisms of the achievement gap. But I'm going to focus here on a different set of brain regions that we also think are really critically involved in explaining the achievement gap. Many studies have found SES-related differences in the structure and function of the ventral visual stream. The ventral visual stream is a set of brain regions located in the ventral temporal cortex that are critically involved in processing of complex visual stimuli. So this is work from Allison Mackey showing differences in cortical thickness in low compared to high SES individuals. And you see where the arrows are pointing to the ventral temporal cortex that there's a large swath of difference, such that children from low SES backgrounds have thinner cortex compared to their higher SES counterparts. And we also see the same thing in Kim Noble's work, looking at cortical surface area, where there's a reduction in cortical surface area in the ventral temporal cortex in low compared to high SES individuals. And indeed, recent work from our lab has found that functional activation in the ventral temporal cortex during a working memory task for faces is associated with socioeconomic status, such that higher SES individuals have higher activation in this region of the occipital fusiform cortex. And recruitment of this region during the task is not only associated with performance on the task, but it's also associated with academic achievement. And so all of this work led us to our current study. And this question of whether ventral visual stream activation may serve as a mechanism explaining the income achievement gap. And to answer this question, we used a sample of 62 children aged 6 to 8 years old. And we used the income to needs ratio as a measure of socioeconomic status. And you can see our distribution here. The children came in for a functional MRI scan. They performed a cued attention task in the scanner. They saw a word on the screen, like the word cup, an arrow pointing to a spot on the screen, and they also heard a word that matched that word that they saw on the screen. They were instructed to pay attention to the location that the arrow was pointing. Then, after a brief delay, they were presented with 4 pictures. On half of the trials, the picture would have appeared in the cued location, and on the other half, it appeared in a different location. And they responded with a button press about whether or not the picture appeared in the correct location. We also had children do a standard test of academic achievement, the Woodcock-Johnson, and we used the academic skills cluster as a measure of academic achievement. So the first question we wanted to ask was whether socioeconomic status is positively associated with ventral visual stream activation. So to do this, we independently defined regions of interest in the lateral occipital cortex and the fusiform cortex. And confirming our previous findings, we found that activation in the left fusiform cortex was positively associated with the income to needs ratio. The next question we wanted to ask was whether academic achievement was associated with recruitment of these regions. So using those same regions of interest, we tested the association between activation and performance on the Woodcock-Johnson. And indeed, we found a positive association for the left and right fusiform cortex, as well as the right lateral occipital cortex with academic achievement. We also confirmed this same result in a whole brain analysis, where we used performance on the Woodcock-Johnson as a predictor of activation during the cued attention task. And we see a positive association with performance on the task in both the fusiform cortex here and also the lateral occipital cortex. And we also see activation differences in the intraparietal sulcus, an important region for attention. So to summarize, what we find here is that the fusiform cortex especially is associated with both socioeconomic status as well as academic achievement. So in this study, we confirm the well-established income achievement gap such that socioeconomic status was positively associated with academic achievement. But given that the left fusiform cortex was also associated with both SES and academic achievement, we conducted a mediation analysis to determine whether activation in this region during the cued attention task mediated the association between socioeconomic status and academic achievement. And indeed, we found an indirect effect of socioeconomic status on academic achievement through activation in the fusiform cortex. And when we account for this, we see that this association is fully mediated and becomes no longer significant, suggesting that activation in the ventral visual stream plays an important mechanistic role in explaining the achievement gap. So to conclude, this study extends previous work showing that visual processing regions play an important role in the development of complex cognitive processes, and that the ventral visual stream functions as an additional mechanism explaining the income achievement gap. So why might childhood socioeconomic status influence structure and function of the ventral visual stream? And what role does this play in the achievement gap? We have recently put forth a model that suggests that these SES-related differences in the ventral visual stream may be driven by lower levels of cognitive stimulation that tend to occur in lower SES households due to time and money constraints. Cognitive stimulation in the context of caregiver interactions may support children's ability to identify and categorize visual stimuli and regulate attention to relevant stimulus features. So for example, a caregiver can highlight the features of an object to help a child learn to categorize stimuli, so such as directing a child's attention to a cat's pointed ears, size, or eye shape as compared to those of a dog. Limited caregiver interactions and reduced access to complex stimuli may constrain the amount of organized feed-forward information that's sent from the ventral visual stream to regions of the brain that support higher order cognitive function. And overdevelopment, this may result in lasting differences in higher order cognitive abilities, and in turn, this may have downstream impacts on academic achievement. And altogether, this may help to explain the income achievement gap. Our current work is focused on characterizing the home environment to understand factors that contribute to this gap and this development, as well as the interplay between the ventral visual stream and prefrontal cortex in supporting development of higher order cognitive function and ultimately impacting academic outcomes. So I want to thank my lab, the Stress and Development Lab at Harvard, all of our funding agencies, and all of you for your attention. Thank you.